This talk will provide an overview of the BioCyc database collection and the Pathway Tool software. BioCyc is both a database collection and a web portal. The databases are encyclopedic references on sequenced genomes and associated metabolic pathways and regulation, with 18,000 total databases for sequenced genomes, of which 60 are curated with their contents derived from 130,000 publications. The BioCyc website provides access to the BioCyc database collection to support tasks such as information seeking for information on a given gene or a given pathway, metabolomics and transcriptomics data analysis, comparative analysis, and metabolic engineering. Pathway Tools is the software used to create BioCyc databases, and it powers the BioCyc website. It's a really extensive toolkit for enterprise pathway genome data management. One of its capabilities is to enable you to install a private set of BioCyc databases locally at your site on your intranet, but in addition, it can create BioCyc-like pathway genome databases from proprietary genome data. Users can edit these pathway databases interactively. The software also performs scientific visualization and analysis. It enables construction of quantitative metabolic flux models using flux balance analysis. It supports all of the same use cases as BioCyc, but it runs as both a desktop application and as a web server. The BioCyc database collection is organized in 3 different tiers, which reflect the quality levels of the databases. The Tier 1 databases are highly curated, in some cases receiving more than a person's decade of curation. The Tier 2 databases are moderately curated, and the Tier 3 databases are computationally derived. All of the databases share the same schema and cover these multiple different types of information, ranging from pathways to proteome to genome. Our curated databases include humans and a number of model eukaryotes, as well as the 2 important model bacteria and a number of other bacteria. The full list of 60, the other missing databases are bacteria. Now, when we create a new BioCyc database, we begin by obtaining the sequenced genome, usually from the RefSeq database. We'll then perform a series of computational inferences, import data from a number of different sources, and in some cases then perform curation, sometimes constructing a metabolic model. The computational inferences include predicting metabolic and transport reactions, predicting metabolic pathways, predicting operons, and computing orthologs, as well as computing PFAM domains. The data import functions import regulatory data, Go terms, and protein features. Now, what do we mean by curation? We mean correcting false positive and false negative pathway predictions, and generally incorporating information from the experimental literature. Curators often author mini-review summaries for genes and pathways. They cite the literature from which they're deriving information. They update many database fields. They add new pathways and modify existing pathways, and they add many types of regulatory information. MetaCyc is the curated multi-organism metabolic database that is our reference database for predicting pathways in sequenced genomes. And so MetaCyc is one database within the BioCyc collection. Unlike other BioCyc databases, MetaCyc does not describe one organism. It's a multi-organism pathway database. Now, the number of genomes here listed here, I'm comparing MetaCyc to KEGG. The number of genomes listed is really the number of BioCyc genomes, and these numbers are about a year old to be consistent with KEGG. So BioCyc contains almost 3 times as many genomes as KEGG does. We have 62,000 publications. KEGG has very few. We have 2,700 metabolic pathways, whereas KEGG has 348 modules, the comparable type of pathway in KEGG. MetaCyc has 16,000 reactions, whereas KEGG has 11,000. KEGG does have more metabolites than MetaCyc does. And MetaCyc's, if we add up all of the MetaCyc mini reviews, it comes to about 10,000 textbook equivalent pages, whereas KEGG has very few of these mini review summaries. Let's now move on to talk about the Pathway Tools software. And an overview article describing all of the capabilities of Pathway Tools is here at Archive. You can download it here. Overall, Pathway Tools enables the creation of multi-use metabolic databases that can be queried via a website or by programmatic database queries. They can be converted into metabolic models, and they can support various types of analysis, such as omics data analysis. The overall workflow for Pathway Tools is to provide an annotated genome as the starting point, and then the pathologic component of Pathway Tools predicts the pathways of the organism and encapsulates those predicted pathways and the supplied genome information as a new pathway genome database for that organism. The navigator component of Pathway Tools can then be used to search and visualize and analyze the database contents. The editor's tools can be used to update the database. And Metaflux is the component of Pathway Tools used to create metabolic models. This slide lists the full set of capabilities of Pathway Tools, which span genome informatics, pathway informatics, regulatory informatics, high-throughput data analysis, comparative analysis, and advanced data manipulation. The genome informatics tools include a number of search operations for gene, protein, and RNA information, as well as a genome site search, a gene information page from which the nucleotide and amino acid sequence can be retrieved, a multiple sequence alignment tool, both a blast search and sequence pattern search capability, the ability to map SNPs to the nearest genes and show their effects on translation, and a genome browser that can zoom from the full chromosome in one page to the sequence level. It has a comparative mode shown here and a tracks facility. It has omics data painting operations, as well as the ability to create a printable genome poster. And it has a circular genome viewer. Pathway informatics tools include search tools for pathways, reactions, and metabolites, as well as information pages for pathways, reactions, and metabolites. Pathway diagrams are generated automatically and can be customized for publication. The software will create personalized multi-pathway diagrams in which individual metabolic pathways can be combined into one diagram, positioned relative to one another, and connections formed between them. And there are comparative pathway analysis tools. The reaction pages will depict reaction atom mappings. And for metabolites, we provide many different search capabilities, as well as to depict all of the reactions, pathways, and regulatory effects of a metabolite. There is a zoomable organism-specific metabolic map diagram, which can be printed as a poster. There's also a metabolic root search tool that computes optimal paths through the metabolic network, and there's a tool for constructing quantitative metabolic models. Regulatory informatics tools will capture regulatory sites and also capture multiple types of regulatory interactions, from transcription factors to small RNAs to allosteric regulation. And this metabolic network diagram is highlighting in red the answer to one example regulatory query, which is to find all the enzymes whose gene is negatively regulated by FNR. And we also provide a regulatory network browser. There's also a Transcriptomics and Metabolomics suite of data analysis tools, including the Omics dashboard shown here, which provides a high-level view of the entire cell, depicting all the subsystems within the cell. And the user can click on any subsystem to drill down into that subsystem. So for example, this biosynthesis panel shows all of biosynthesis divided into amino acid biosynthesis, nucleotide biosynthesis, carbohydrate biosynthesis. The colored bars are showing different time points in this experiment, and the fat dots are showing the averages of all the gene expression levels in that time point, so that we can see which systems are activating, which systems are decreasing their activity very clearly. and the user can click on any of these subsystems to see the details of what's happening at the gene level. We also have tools for pathway visualization of omics data that will paint omics data onto individual pathway diagrams, pathway collage diagrams, onto the zoomable metabolic map diagram, and onto a table of pathway diagrams sorted by an activation score that we compute for each pathway. There's also a classical enrichment analysis capability, and we can visualize transcriptomics data on the genome map and on a regulatory network diagram, and we can compute what are called pathway covering sets for metabolomics data. Now we have a few new pathway informatics tools. We have a Metabolic Network Explorer tool that enables the user to interactively explore the metabolic network by traversing a set of related compounds of interest. So for example, we always start at a given metabolite. In this case, we're starting at L-arginine in the E. coli network. And this graphic here indicates the two main metabolites that are reactants of reactions that synthesize arginine, and the four main metabolites that are products of reactions in which arginine itself is the input. Now at this point, I can click on any of these plus signs to expand our evolving path in that direction. Here I've clicked on agmatine, and so we see this reaction, and we see that agmatine can further lead to both putrescine and urea. And I'm going to click on putrescine to go to this screen. So we're now slowly building up this pathway. And as you can see, putrescine can use additional metabolites. I'm going to click on spermidine, and we get another step in our diagram. And at any point, I could start moving backward through the network, or move forward from another point like agmatine, etc. There are also controls for customizing the diagram. So here I've removed the EC numbers that were there previously, and I've added chemical structures. We can also save the diagram and restore a saved diagram. Now another recent improvement is that we've re-engineered the Pathway Tools metabolic network diagram to give fast interactive semantic zooming. The diagram now supports multiple cell types, and we've extended it to microbial communities. This tool produces organism-specific metabolic map diagrams that are organized so that we have biosynthetic pathways on the left, catabolic pathways on the right, energy metabolism down the middle, and individual reactions not part of any specific pathway on the right side. Transporters are shown in the cell membrane, and periplasmic reactions are on the right here. Now, if I switch over to a web browser to look at the live version of this diagram, which we can get through the Tools menu by clicking Cellular Overview for whatever is our currently selected organism, which we can change using this button. This is the initial version of the diagram, and I can move the diagram around. And by rotating my mouse wheel, I can zoom in on any region of the diagram. In this case, I'm zooming in on the cofactor and vitamin biosynthetic pathways in E. coli. And we'll begin to see metabolite names. Then we'll see pathway names. We'll see gene names and enzyme names appearing. And we can further drag the diagram around. And we can also search the diagram using these tools on the right to highlight, for example, a gene or a metabolite by exact name or by substring. I'm going to search for L-arginine, and we can see it's come up in a number of places in red. That is an arginine acid resistance pathway. We can see here we have an arginine degradation pathway and another arginine degradation pathway. We have some arginine transporters up here. And we can also overlay high throughput data sets onto this diagram. So for example, if I click Upload Data from File, I'll get a diagram such as this one. Now it's really the same diagram, but each reaction is colored with a color indicating the expression levels loaded from this tab delimited file. Now in the specific file that we've just loaded, E. coli start out growing in anaerobic conditions, And then we have 6 different time points during their shift to aerobic growth. And what you'll see is that most of this diagram is gray because a few extreme values are throwing off the scale. And you can see actually that there are just very few data points up in the upper region. So what I can do is to adjust the color scale to give some more range to the data. So you can now see we're getting a lot more range in what we're looking at. Here we're just looking at the first data set, the first time point in this of the 6. If I click this button, it will begin to play through these different time points. And we can also change the speed of the plane. We can stop the animation at any time. So if we see here, we can see that fermentation is turned on quite strongly and TCA is largely turned off. But as we go to the later time points, here TCA is turned on very strongly, whereas fermentation has mostly turned off, although these new branches are turning on. We can also continue to zoom in, just as we could for the unhighlighted diagram, to see which metabolites are which. We can find out what that reaction is. Everything has a tooltip in this diagram, both the reactions and the metabolites, to identify them. And if we mouse over that, if we click on the Omics button, we get an Omics pop-up that graphs the levels of the genes on that reaction. And similarly, we can click on this one to graph its levels. Now, some other improvements that we've made to this diagram are to give it the ability to depict cell type subsets of metabolism. So in this case, we've used a human proteomics data set to define which subsets of the metabolic network are active in different human cell types. And you can see that we've grayed out regions of the human metabolic network that are not present in this endothelial cell type, as well as transporters that are not active there. And then here we've extended the diagram to work with microbial communities. Here we've created pathway genome databases for each of the 8 members of the altered Shadler flora, which is a mouse microbiome, gut microbiome. And we've extracted the cellular overview diagrams from each of the 8 organisms. We've actually highlighted within them the pathways that are unique to each organism. And we're also depicting metabolites that are exchanged between the different organisms. So we can use this diagram to interrogate the metabolic interactions between these organisms. And this will lead me to other microbiome analysis capabilities of pathway tools. In general, the software can create pathway genome databases for a whole set of genomes from a microbial community, such as if we have metagenome assembled genomes extracted from a community. We can use a batch mode of pathway tools to set up all of the mags. Each mag will go in a different folder, and then the software will create a new PGDB from each folder. Once that's been done, we have new multi-search capabilities that let us search across a whole community of PGDBs to find which organisms have a given gene, which organisms have a given pathway, or which organisms have a given metabolite. We also have tools for assessing the metabolic complementarity among organisms, such as to find which organisms can nourish or compete with a potential therapeutic organism. We also have a tool for multi-organism route search that computes an optimal route through the metabolic network from a starting metabolite to an ending metabolite via reactions obtained from multiple metabolic networks specified by the user, such as the multiple organisms in a community. The tool can also create metabolic models for multiple organisms in a community and combine them to simulate that community. And we can create multi-organism metabolic map diagrams for omics data analysis, as I showed you in the previous slide. We have a number of new genome informatics tools, but I'm first going to review some of our previous tools. The genome browser can depict an entire genome in one screen. This is the entire E. coli genome in one screen, while a little bit is scrolled off the bottom. Each arrow is one gene, and adjacent genes in the same color are in the same operon. We can zoom in a level to see in this case, the region of the genome right around in here, at a slightly higher level of magnification. If we zoom in more, we'll see each gene very distinctly, along with promoters and terminators. We can make out pseudogenes and RNA coding genes with this different arrow type here. And if we zoom in further, we'll see the sequence level. Both the nucleotide and the amino acid sequence of this gene are depicted. This is the genome browser in its comparative mode, where we've lined up the chromosomes for several related bacteria, all centered around a single gene and its orthologs in those organisms. And in this version of the diagram, the colors depict orthologs. So all the genes in green are orthologs of one another, and all the genes in orange, etc. So we can see some differences in genome organization with this gene inserted in this genome. In this genome, we have two pseudogenes that are not present in the other genomes. And we have a new circular genome viewer that lets the user define a number of concentric tracks on which different information is plotted. On the left here, the black outer track shows pseudogenes in E. coli. Then transcription factor binding sites are shown in red. In blue, we have rep elements and transposons. And in purple, we see cryptic prophages. In this version of the diagram, we're showing the binding sites for a number of different transcription factors from ARC-A, FNR, CRP, and LEX-A. We've also added new sequence alignment tools. Alignments are now computed with +del omega, and we've added MSA viewer as the viewer for multiple sequence alignments, as shown here. Let's now consider the regulatory informatics tools within Pathway Tools. We have a diagram called the regulation summary diagram that depicts all the regulatory influences on a gene and its product, from transcription to translation to post-translational regulation. For the TRPA gene, we're seeing the TRPR transcription factor regulates its transcription, whereas attenuation occurs at the translational level. And finally, PLP activates the final enzyme. For MAB, there's no transcriptional or translational regulation, but there's a lot of post-translational regulation by these different metabolites. In addition, on the gene pages for transcription factors, there's a Regulon tab that will list all the different operons that the gene regulates, with the binding sites for that regulator depicted. Regulatory sites are also shown in the genome browser. Here we have several activator sites upstream of this gene and inside this gene. We have a combination of activators and repressors upstream of this gene and upstream here, and still more binding sites down here. Now we also have a regulatory network diagram called the regulatory overview that consists of 3 concentric ellipses. The inner ellipse depicts the global regulators in this organism, transcriptional regulators and small RNAs, and the middle ring depicts other regulators. The outer ring depicts genes that are not regulators, and we've clustered together all genes that are regulated by the same set of regulators will be shown in one of these clusters. For example, one of these large clusters are all the genes in E. coli regulated by RPOD. Now I can right-click on any of these genes to ask to see all the genes that regulate it or all the genes that it regulates. And so for LEX-A, we're now seeing all the E. coli genes that it regulates. And I've further clicked on one of them to show the genes that it regulates. Let's now consider one of the advanced data manipulation tools provided by Pathway Tools, which is called SmartTables. SmartTables enables the user to collect and save lists of database objects. It could be lists of genes or metabolites or pathways, etc. The user can explore them interactively, combine them through set operations, filter them by removing items that match a given search string, and transform them into related objects. So for example, we can transform a gene list into a pathway list. The user can also share SmartTables with the public or with specific collaborators and can publish them online as well, and they support omics analyses. Here, for example, is a gene list, a gene smart table, where the first column lists the gene names, and then the other columns list various database attributes. So for example, this column lists the product of each gene, and this lists the pathways of each gene. To define a new smart table, we can import the list of objects from a file, or we can create the object from query results from a search that we've performed. Some other examples, here's a metabolite smart table where we've added a number of columns showing links to other databases. And for this metabolite smart table, we're showing some other attributes from the database, namely molecular weight and monoisotopic molecular weight. We can also show the structure of the compound and its SMILES string. Back to a gene smart table, we're now showing an accession number, the genome coordinates of the gene, and the nucleotide sequence can be shown in the smart table. And then transformations allow us to compute a new column based on computed relationships within the database. For example, we can take a metabolite smart table, transform it into a set of pathways, and then into a gene path smart table for all the genes in each pathway. We could also create a smart table containing a transcriptional regulator and transform that to all of the genes regulated by that one regulator or by a set of regulators. We can also transform a gene smart table into a smart table of all the regulators of those genes. We can transform a gene smart table into a list of all the transcription factor binding sites that control those genes, and from there into a list of sequences. We could create a smart table of nucleotide positions and then transform that to the closest genes and paint it onto our metabolic overview. So really, smart tables often let biologist users perform analysis tasks that they'd otherwise need programming skills to perform. And here's a visual example where we've started with these metabolites, transformed them to reactions involving those metabolites, pathways involving those metabolites. So this metabolite is involved in these two pathways because this one wraps around. And then these two pathways were transformed to their gene lists. So these are all the genes, human genes, involved in these pathways. Now let me summarize the availability of both BioCyc and pathway tools. The BioCyc website and database files are available via subscription, where subscription revenues help support our curation efforts. But because the ECCB and MetaCyc databases, because their curation is supported by government grants, those databases are freely available to all. And this is a good way to explore BioCyc to find out its capabilities, is to explore these two databases. The Pathway Tools software is free to nonprofits, and we charge a fee to commercial organizations. And it's available for Mac, Windows, and Linux. And you can obtain Pathway Tools here. And I'm happy to take questions.